All right, so now let's continue with the second part of lipid chemistry. For this lecture, we shall be discussing the more complicated lipids, such as the phospholipids, the sphingoglycolipids, cholesterol, steroids, and then we'll end the lecture with lipoproteins. Let's just have a brief recap of what we have discussed earlier. So we all know that lipids are large heterogeneous class of biomolecules. They are grouped together because they are characterized by low water solubility and high solubility in nonpolar solids. The first member of the lipid family we discussed earlier are the fatty acids. These are hydrocarbons with hydroxyl, or with rather carboxyl functional group. Well, fatty acids can be either saturated or unsaturated. And the storage form of fatty acid is the triacyl glycerol. So these are fatty acids sterified to a glycerol backbone. So now let's move on with phospholipids, the next member of the lipid family. In contrast to the first lipids that we have discussed, which are almost exclusively hydrophobic, phospholipids are amphipathic class of lipids, which means these are lipid with a polar head and a hydrophobic tail. Now, both components are attached to a backbone. One of the important functions of the phospholipid is that they are important component of the cell membrane. Well, there are two kinds of phospholipid. Glycerophospholipid is the first one. Here, glycerol is the backbone. And the other kind of phospholipid is the phosphosphingolipid, where sphingosine is the backbone. Well, let's discuss first the glycerophospholipid. So this is a schematic diagram of a glycerophospholipid. It is composed of a polar head and two hydrophobic tails. So the backbone, as I have said earlier, is a glycerol. Bonded to it is a phosphate. And this is what is responsible for its hydrophilic or polar property. Now, two fatty acids are bonded to the glycerol backbone. So the fatty acids can be either saturated or unsaturated. And the tail of the fatty acid is what gives it its hydrophobic property. So we have here a schematic diagram of the structure of a glycerophospholipid. So just like what I said earlier, the backbone is glycerol. Sterified to it are two fatty acids, which may be either saturated or unsaturated. And the head, of course, is a phosphate. And there may be some other polar components attached to the phosphate head. So the structure of a glycerophospholipid is shown here. So highlighted in blue is the backbone. And this backbone is called glycerol. So it's a 3-carbon alcohol. Sterified to it are two fatty acids, the one that gives it its tail. And the head, of course, is a phosphate. And to the phosphate, we may add other components that will give rise to the different types of glycerophospholipids. So what are the different classes of phospholipids? So here are some examples. Well, the simplest is called a phosphatidic acid. The head is nothing more than a phosphate. Well, another kind of a glycerophospholipid contains an amino acid called serine, as shown here. So this glycerophospholipid is known as phosphatidyl serine, a very important component of the cell membrane. Now, an amino alcohol called ethanolamine can also be bonded to the phosphate head, giving rise to another type of glycerophospholipid. This is called a phosphatidyl ethanolamine. A derivative of ethanolamine is called choline. So it also contains an amino group. Bonded to it are 3 methyl group, it's also an alcohol that is esterified to a phosphate. Now, just like phosphatidylserine, phosphatidylcholine is also an important component of the cell membrane. And then we can also bond a glycerol alcohol to the phosphate, giving rise to another kind of a glycerol phospholipid. This is what is called the phosphatidylglycerol. As I have mentioned earlier, the other backbone for a phospholipid is a sphingosine, shown here. So it's a long molecule composed of 1, 2, 3, 4, 5, plus 12, 17, 18 carbon. It contains hydroxyl functional groups, it's an alcohol. It also contains amino group, so it's an amino alcohol. To the alcohol, we attach a phosphate plus a choline. This is responsible for its polar property. And to the amino group, we bond there through an amide linkage a fatty acid, and this fatty acid is what would give it its tail. Let's compare now the two phospholipids, phosphosphingolipid and glycerol phospholipid. So on the top panel, we have the phosphosphingolipid, whereas on the bottom panel, we have the glycerol phospholipid. So what is their difference in terms of structure? Of course, a phosphosphingolipid contains sphingosine as a backbone, highlighted in violet, whereas a glycerol phospholipid contains glycerol as its backbone. So it's much shorter, so it's a 3-carbon alcohol. Well, both of them contains a fatty acid that will serve as a tail, but for the sphingophospholipid, it only contains 1 fatty acid. Whereas for glycerol phospholipid, it contains 2 fatty acids. So the head is the same. The head is composed of phosphate, only shown here as an X. Whereas here we see phosphate also forming the head, and to the phosphate we can also add other components such as an amino alcohol. So to recap, the difference is in their backbone. This is a sphingosine backbone, whereas here the backbone is glycerol. There is only 1 fatty acid for the sphingophospholipid, whereas there are 2 fatty acids for the glycerol phospholipid. They also have the same head, phosphate, shown here only as an X. Like what I said earlier, phospholipids form the lipid bilayer in cell membrane. As you can see in the orientation here, the polar head are oriented towards the aqueous environment, whereas the non-polar tails are oriented towards each other. Now take note that the composition of the outer leaflet is different from the composition of the inner leaflet. We call this asymmetry. The outer leaflet is predominantly composed of phosphatidylcholine. On the other hand, the inner leaflet is predominantly composed of phosphatidylserine. Now, if phosphatidylserine happens to flip into the outer leaflet, this is actually a sign that the cell is undergoing what is known as programmed cell death. Now, the degree of saturation of the tail would also affect the physical property of the cell membrane. When the cell membrane is composed of phospholipids that contain exclusively saturated fatty acid, we can see that the lipids are more compact to each other. Thus, the cell membrane is less fluid. On the other hand, if we have phospholipids that contain unsaturated fatty acids, this would result to a kink on the tail. As a consequence, the molecules are farther apart. This would give now the cell membrane its more or less fluid character. Now let's move on to sphingoglycolipids. So this is somewhat similar to a phospholipid, but this is exclusively found in nervous tissue. So what are sphingoglycolipids? So these are a class of lipids composed of sphingosine backbone. They are also sterified. The backbone is sterified to a fatty acid, but the head, rather than a phosphate, is a sugar, either a mono or an oligosaccharide. So we have two examples here, cerebroside and ganglioside. So cerebroside is the simplest of the sphingoglycolipid. So the head is usually a monosaccharide, either a glucose or a galactose. On the other hand, sphingoglycolipid called ganglioside is more complex because the head is composed of oligosaccharides. So these are composed of up to 7 monosaccharides that are linked together. So we have schematic diagram of the structure of a sphingoglycolipid shown at the bottom panel of the screen. So let's take a look at them. So this glycosphingolipids contain sphingosine as their backbone, shown in green. They also contain fatty acid, usually a saturated fatty acid such as stearic acid. But what makes ganglioside different from cerebroside is the polar head. A ganglioside contains a string of monosaccharides linked together. So this is what we call an oligosaccharide. Whereas a cerebroside only contains one sugar. We call that a monosaccharide. That sugar is either a galactose or a glucose. So let's start first with the cerebroside, the simplest of the sphingoglycolipid. So usually they have a glucose or a galactose as a carbohydrate unit. So the diagram of the structure is shown here. Like what I said earlier, the backbone is sphingosine, sterified to the amino group, is a fatty acid, a saturated fatty acid such as stearic acid. And the head is a sugar, a monosaccharide. In this case, the monosaccharide is galactose. So this cerebroside is also called galactosyl ceramide. So this is the principal glycosphingolipid in the brain. As a matter of fact, they are present in all nervous tissues and can compose up to 2% of the dry weight of gray matter and 12% of white matter. So they are actually the component of the myelin sheath. In other words, the myelin sheath is composed of galactosyl ceramide. Let's compare now a phosphosphingolipid and a sphingoglycolipid. So both of them are amphipathic. In terms of function, a phosphosphingolipid makes up the cell membrane in all tissues of the body, whereas the sphingoglycolipid is only found in nervous tissue. Now, what do they have in common in terms of function? Both of them contains this as a backbone. And what do we call this backbone? This backbone is called sphingosine. What else do they have in common in terms of structure? Sterified to an amide linkage is a fatty acid that forms a tail, a saturated fatty acid such as stearic acid. So they have this in common. What makes phosphosphingolipid different from sphingoglycolipid in terms of structure? Well, it's the head. Sphingophospholipid contains phosphate as a head, and bonded to it is choline. Whereas here, the head, of course, is a sugar, either a galactose or a glucose. In this case, it is a glucose. Let's now compare all the lipids that we have discussed so far. And this would include triacylglyceride, phosphosphingolipid, and glycerophospholipid. So let's discuss first the storage lipid, and that is the triacylglycerol. We all know that the backbone is glycerol. Sterified to it are 3 fatty acids. And then there are the membrane lipids, the phospholipids found all over the body that makes up this cell membrane. And then we have a lipid, a membrane lipid exclusively found in the nervous tissue. We call it the glycosphingolipid. So let's discuss first the phospholipid. There are 2 kinds depending on the backbone, the glycerophospholipid and the phosphosphingolipid. And obviously the difference is in terms of the backbone. Glycerol for glycerophospholipid, sphingosine for phosphosphingolipid. What do they have in common? The tail. But we have 2 fatty acids bonded to glycerol here, and only 1 fatty acid bonded to the sphingosine. Now what about the head? Both of them contains phosphate as their polar head, and bonded to the phosphate is an amino alcohol, such as choline. Now, similar to the phosphosphingolipid, but is exclusively found in the brain, is the glycosphingolipid. So how is it similar? Well, the backbone is sphingosine. Sterified to it is a fatty acid. But what makes it different? The head is a sugar, either a monosaccharide or an oligosaccharide. Now let's move on to cholesterol. So the lipids that we have discussed previously are predominantly linear in terms of structure. Now what makes steroid different from the previous lipids that we have discussed is that its structure is based on a ring, a 4-ring system shown below. We call this the cyclopentanopenanthrin ring. So all steroid derivatives, such as cholesterol, would possess this particular ring. Well, 3 of the rings, such as A, B, and C, are 6-membered, fused together, while the 4th ring is a 5-membered ring. So this molecule has diverse role throughout both the plant and animal kingdoms. Let's discuss first cholesterol. So this is the most abundant steroid in the human body. So the ring is shown here. We call this a cyclopentanopenanthrin ring. And a derivative of a steroid that contains that ring is the cholesterol. So it also contains that ring. It is an alcohol because it contains a hydroxyl group at position number 3. It is an unsaturated alcohol because it contains a double bond between carbon number 5 and number 6. It contains methyl side chains in number 10 and number 13, plus another side chain located at number 17. So cholesterol is actually an unsaturated alcohol that possesses the cyclopentanopenanthrin ring. Well, much of the cholesterol is sterified to fatty acids. As a matter of fact, we store cholesterol sterified to fatty acid. When they are free, when they are unsterified, they tend to form gallstone. And like what I said earlier, this is the most abundant animal steroid. As a matter of fact, a 60-kilogram person has a total of about 175 grams of cholesterol distributed throughout the body. So what is the function now of cholesterol? Well, cholesterol has a very bad reputation because we always associate it with atherosclerosis and coronary heart disease, but it does have important physiologic functions. And one of that is that it is a minor component of the cell membrane. So phospholipids are still the major component of the cell membrane, and cholesterol is a minor component. And it's important because it helps to keep the cell membrane fluid. Another important function of cholesterol is that it is a precursor for the synthesis of all other steroids, such as sex hormones and vitamin um, D, as well as vitamin A. So like what I said earlier, cholesterol is a minor component of the cell membrane. So other components of the cell membrane are the phospholipids, peripheral proteins, which may be attached to a carbohydrate chain. So the cholesterol molecule are intercalated in the hydrophobic tail. So they keep the cell membrane fluid. That means that they prevent the cell membrane from crystallizing when the temperature is low. So it prevents the cell membrane from becoming too hard when the temperature is low. It also prevents the cell membrane from becoming too soft when the temperature is high. So in other words, cholesterol maintains an acceptable level of membrane fluidity. So to recap, the function of the cholesterol in the cell membrane is to add firmness and integrity to the plasma membrane. So it helps maintain an acceptable fluidity. In other words, it prevents extremes. It prevents the cell membrane from becoming too fluid when the temperature is high, or too hard, or too firm, or too solid when the temperature is low. So cholesterol is also very important because it's a precursor material for the synthesis of all other steroids. So this includes sex hormones such as testosterone, estradiol, and progesterone, adrenal hormones such as cortisol. So we are just going to enumerate for now the other hormones that can be produced from cholesterol. We shall be discussing the biosynthesis next semester, if you're still around. Cholesterol is also the precursor for vitamin D as well as for vitamin A. And finally, cholesterol, when degraded, will give rise to bile salt and bile acids, which are important for emulsification during lipid digestion. So just to illustrate, cholesterol is the precursor for the production of important hormones, such as the sex steroid hormones, progesterone and androstenedione, as well as adrenal hormones like cortisol. So like what I said earlier, cholesterol is also the precursor for the production of vitamin D. And this happens in the skin. So we won't be discussing how vitamin D is converted into cholesterol. We shall reserve that for next semester, again, if you're still around. So the difference is that if you take a look at cholesterol, it contains 4 rings, whereas for vitamin D, almost the same structure, except that the second ring is now open. It only contains now 3 rings. And this happens simply by UV light exposure of cholesterol. Now, the degradation product of cholesterol is the bile salt or bile acid. So even when it is degraded, the ring called the cyclopentanopenanthrin ring is still maintained. And if you notice, it contains a carboxyl functional group. That this is the reason why it is referred to as an acid. Now, this could possibly the attachment site for either glycine, amino acid glycine, or taurine. And when those amino acids are added, we now refer to it as a bile salt. Now let's discuss the dietary recommendation for lipids. So The limit for total fat intake should be less than 30% of your total caloric intake. So for example, if you're on a 2000 kilocalorie diet, 30% of that would give you 600 kilocalories. Simply divide it by 9 because one gram would yield 9 calories. In a day, you're only allowed to have 65 grams of fat. And saturated and polyunsaturated fats should make up less than 10% of your calories. And what about cholesterol? How much cholesterol are you allowed to have in a day? So in a day, you're only allowed to have less than 300 milligrams per day. To put that into context, an egg contains about 200 milligrams of cholesterol in a day. So in a day, you're only allowed to have one egg, because if you have 2 or more eggs, you would now exceed the 300 milligram cholesterol limit. And finally, I will be introducing you to lipoproteins, which again will be discussed extensively in lipid metabolism next semester. So these are clusters of not just lipids, but lipids and proteins, and their function is to transport fats. So they are used as transport vehicles for lipids. So there are 4 classes of lipoproteins: chylomicron, very low density lipoprotein, low density lipoprotein, and high density lipoprotein. Well, the typical composition of a lipoprotein is shown here. So the core is where you're going to find the most hydrophobic component. This includes triglycerides and cholesterol esters internally. The proportion of triglyceride and cholesterol depends on the type of lipoproteins. Now, it's covered externally by a single layer of phospholipid, and embedded on the phospholipid monolayer are the proteins, referred to as the apoproteins. Let's take a look at the relative sizes of the lipoproteins. So as illustrated here in violet is a chylomicron, so it's the largest of all the lipoprotein. Next in terms of size is VLDL, followed by LDL, and the smallest is LDL. And why is this so? Why is chylomicron the smallest? Let's take a look at their relative composition. Well, chylomicron is big because it contains a lot of triglyceride, dietary triglyceride, and very few proteins relatively. This is the reason why it's huge, but it is very light because it contains predominantly triglyceride. On the other hand, for HDL, although it is the smallest, it is packed with a lot of proteins. And this is the reason why it is very dense. In terms of composition, chylomicron and VLDL are very similar in that they contain a lot of triglyceride. Now, if you take out the triglyceride in VLDL, this VLDL is converted into LDL. So in other words, LDL is simply VLDL minus cholesterol. So now we have reached the end of our lecture. Let's just summarize what we have discussed. So phospholipids are amphipathic. They may have either a glycerol or a sphingosine backbone. The next lipid that we discussed today are the cholesterol molecule. Is a cholesterol molecule. It possesses the cyclopentanopenanthrin ring. And there are important derivatives that can come from cholesterol. This includes steroids and vitamin D. Finally, the most complex of all the lipids are the lipoproteins. So these are complexes composed of lipids and proteins, and their function is to transport lipid. And the different classes of lipoproteins include chylomicron, VLDL, LDL, and HDL. So that concludes the lecture. And if you have any questions, then we can answer them during the enhancement question and answer session that we shall be having on Monday. See you then.